To all, myself Dr. Ketan Shah, Professor, Parul Institute of Pharmacy and Research, Parul University, welcomes you in this webinar on co-processing in pharmaceutical excipients. It is an immense pleasure for me to welcome and introduce today's speaker, Dr. Jaideep Patel. Dr. Jaideep completed his BPharm and MPharm from Bapuji Pharmacy College, Davangere, Karnataka. Dr. Jaideep completed his PhD from M. Chandracharya, North Gujarat University, Patan. Dr. Jaideep is having 14 years of teaching experience. He has passed GPSC exam in 2006 and joined BK Modi Government Pharmacy College, Rajkot. Dr. Jaideep Patel is working as a Class 2 Senior Lecturer GES BK Modi Government Pharmacy College, Rajkot. Previously, he worked at Cologne Institute of Pharmacy, Cologne and Bavaria Institute of Pharmacy, Vadodara, Gujarat. He is approved PG guide at GPU. He has published many research papers in various international and national journals. He has also presented many oral and poster presentations at various conferences, workshops, and symposiums. He is approved external examiner at UG and PG courses at various universities of Gujarat. He has also received award for Best Journal Sector Officer from Rajkot District Election Office in 2018 and 2019, both consecutively years during part of national duty on behalf of Election Commission of India, New Delhi. He has delivered various expert sessions at various pharmacy institutes across Gujarat. So, with this brief introduction, I would like to request Dr. Jaideep Patel to please deliver this session. Over to you, sir. Thank you.
Okay, thank you, Dr. Ketan. So, good morning, all of you. Today on 26th May, we are going to attend the webinar on co-processing in pharmaceutical excipients. So, all the I may fulfill your expectations, whatever you need from this topic. I will try my best to convey the whatever you wish in from this particular co-processing in pharmaceutical excipients. Basically, we know very well what are ongoing in COVID era. Many pharmaceutical drugs are having a problems in their biopharmacokinetic, physicochemical properties, all etc. different states. Many drugs have a problems in their absorption and problem in their delivery, etc. So basically, that today whatever we are discussing co-processing is also that is one of the part that can you can achieve many things from this particular co-processing area. So basically, that co-processing of pharmaceutical excipients that we are going to present, particularly for excipients we are preparing in that way that can reduce economy of the particular product. Right. So basically, if we see the history of this co-processing, so in 1977, that Joseph Fagin has started this co-processing while he was using the coal and oil crude that normally tire with the coal that getting the crude oil. They were the crude oil, which was a high hydrogen content. So this co-processing started from basically that advantage with advantage, right? Then later on that cement production also they are using a co-processing. The hazardous waste management. This is a also that is a very big area, or you can say that is a lot of opportunity for business in this area. If we basically that particularly we see that waste material, solid waste management. So that can solid waste management that is normally particularly that can be basically co-processing you can achieve in that waste management. Then particularly steel industries also that is steel way in that also that is co-process is widely used in pharmaceuticals that is widely used in co-processing. If you see in pharmaceuticals, so in 1988 that was started with microcrystalline cellulose and calcium carbonate was co-processed first.
Co-processing. Am I audible, Ketan sir?
Yes, sir. Yes, sir. Now you are
Co-processing
audible.
is basically for cellulose and lactose also, that is normally uh, carried out in 1990, right? Then co-processing, that is glucomannan and galactomannan in 1996 was carried out in that area. Next, Ketan sir. So what is co-process? First, we need to understand the definition of co-processing. What is exactly co-processing? That co-processing means we can consider that is the combination of two or more compendial or non-compendial, means pharmacopoeial or not pharmacopoeial, means that is approved, newly approved or already accepted. Such excipients that designed to physically modify their property. We are going to physically modifying them only. We are not particularly that chemically any processes we are carrying out. Only just physically they are modifying, right? So their properties in manner not achievable by simple physical mixing, right? Sometimes you need a spray drying, sometimes you need a wet granule, different methods you need for physical mixing, basically. So without significant chemical change, you have to obtain this co-process excipients. Basically, we can say that is the necessary requirement is there should not be form any covalent bond. There should not be form any covalent bond in the new co-process excipients, right? Because it is if it is able to form a co-process excipients, then it is able to form a new covalent bond. That means it can interact with anyone. So that is rejected. In the particular co-processing, you just remember there should not be any covalent bond between the newly formed co-process excipients. So that should be only a physical change. We studied in a unit operation. Unit operation means only physical changes, and unit processes means on physical changes as well as chemical changes. So here we need to only focus on the particularly physical changes. There should not be no chemical change in the structure. However, some instances that formation of necessary component may occur in the in situ salt form. Sometimes it forms a in situ while on standing or on shelf, or sometimes it forms a inside when reaction is going on. Next, sir. So basically the co-process excipient composition profile, if we decide, then ratio is very much important. First of all, we need to identify the two different co-excipients. What should be the co-excipients? What should be the individual excipients? We need to identify their profiles, both the excipients. What are the particular this excipient is showing example disintegration good, this showing particle size good. Likewise, you need to identify the properties of individual excipient first. Then you need to decide, you need to select the excipients based on the experimentations or preliminary trials, that this and this excipient should be co-processed. And once you decide these two excipients, then what should be the ratio for this co-processing, right? So that all those things you need to understand, and in that way you need to particularly mix the both the excipients, and you have to co-process them with suitable methods, and go for the all the evaluation characters they passed for the new moiety, new excipients moiety, right? So that basically that performance of in that performance should not be intended, right? Not impact the intended performance. So there should not be any changes in their intended whatever we desire performance from both the excipients by co-mixing. That should not be not impacted. Next, sir. So for before co-process excipient, we need to understand we should have a analytical method development first. We should have a good analytical method which can measure everything, right? So while we will form a co-process excipient, so there are different various methods. So according to IPEC, that is a normally international pharmaceutical excipients that says that normally elemental impurity should not be there in the in the co-process. So analytical method should be validated and reproducible in this case. Easily they can identify the elemental impurities, so that can degrade the co-excipients in future on shelf life. Then we can consider a residual solvents. There should not be any traces of the residual solvent in the co-process excipient. Otherwise, on standing or shelf life, there are uh, uh, chances of interaction between the drug or excipient. If any residue is present in excipient, if we are preparing any dosage form, they can might be affect the structure of the drug, and that instability problem starts. So the co-process excipient should be in that way. You can measure the residual solvent present in the co-process excipients, or there should not be no residual solvents present in the co-process excipient. So for that purpose, you need to develop a analytical method. Viscosity is also affect in that case on storage, or there's particularly we can say that is a shelf life can be changed if not proper viscosity is maintained. So analytical method should be developed for that maintaining the viscosities, and that particle size measuring is very much important. So method should be developed in that way you can measure accurate particle size. Degradation, if any degradation happens with the two co by particularly when we are formulating a formulation, so degradation should not be that measured by the analytical method. Limit test, that different limit test normally carried out, and then water content also identified. If we see that co-process excipients, normally what is co-process excipient? So if we see co-process excipients, basically that stability is very much concerned for the co-process excipient. So basically that co-process excipient stability, that is very much important. So for that purpose, their light photo stability or their long-term stability can be evaluated by using the particularly with the packaging and container also, that should be evaluated. Basically whatever packaging material we are using for the dosage form, that should be particularly stable with the excipient stability. If we see that risk analysis, that after checking the packaging material and or everything, that co-process excipient that can be carried out by quality by design actually. So initially risk assessment can be identified. For that purpose, you need to identify different parameters according to the ICH-Q9, that whatever we particularly discussed earlier. So basically that control and then review, that is according to QBD method or risk analysis we have to perform, and our formulation should be a properly. In that case, that should not be any changes. Whatever we desired, we require a desired property, right? Next, sir. So what should be the novel co-process excipients and patient? What about the patient safety? So whatever novel co-process excipients we are preparing, that should have a safety data required. Proper safety data is necessary. So you can see the that normally inactive ingredients database excipients are there. That is a degree of novelty is higher. But what happened? The amount of safety data is also very much required. So as the formulation is changes, that you need to concern about the patient safety always. you need to concern about the patient safety is first, right? Next, sir. So if we see IPEC history and guidelines, that normally we have to follow the different safety evaluation scheme that is given by IPEC. That is normally different forms are there, IPEC forms that you need to fulfill and submit to the IPEC. That is next, manufacturing of co-process excipient. Next, sir. Manufacturing of co-process excipients and process robustness. So those who, whatever method we are selecting, that should be consistent, that can form a consistent co-processing excipients with the same quality, same good quality, same quality that whatever we required. So manufacturing process should be validated. So continuous, all the batches should be same performance, should have a same performance in that all the cases whatever we desired. So manufacturing, if we see, so consistent method, and also the yield is also very much concern here. So if manufacturing at large scale size, so that should be in that way, so that yield also that is properly can be obtained, as well as economical. We can say that cheapest method should be developed for the manufacturing of the co-processing. Otherwise, simple excipients will be performing, and same performance we are getting from the co-process excipients. So there is a much more costlier than the simple excipients. So their robustness is very much important. Process validation, everything is much more important. What are the advantages of co-process excipients? The synergetic effects can be obtained. Single excipient with multiple functionalities can be obtained. Like one excipients have a more than different characters like compressibility, flowability problem, solubility problem can be achieved. Overcome the disadvantages of sometimes some single individual components. Then dilution potential can be improved by using sometimes co-processing. Improve the physicochemical properties like a particle size and all that can affect our uh, particularly different formulation factors. Next, so what are the functionalities? What are the different functionalities of the co-process excipient? So basically we achieve a mixture, improved mixture by getting a surface structure changes, by granulation, different granulation method, by altering a particle size. You can get a flowability, good flowability of the particle. Then increase in particle size, sometimes uh, crystal states, changes in crystal state, surface structures that gives a good compressibility, right? Then surface porous structures, moistures content, which sometimes gives a good improved disintegration also. So many more advantages are there by the co-processing. Some of the marketed co-processing, marketed co-process excipients are there. So you can see that advantage FS, that by fructose and starch they made. Basically, such different advantages are there for the different marketed co-processing excipient. There is very large market, and by 10 years it is a growing area where company will desire a direct compressible or directly use. You can use a direct by reducing many steps. You can using the co-processing excipient. Then different methods of preparations are there for the co-processing. Solvent evaporation technique is there. Hot melt extrusion is there. Co-milling, co-precipitation you can go. Spray drying method is there. Which one is the best method considered? Wet granulation method, dry granulation by roller compactor, and co-crystallization. So these are the different methods you can adopt for a preparation of co-processing. This is one of the patent from our college student, that Puneet Rathod has filed the patent on the co-process pharmaceutical excipients. So we will look at what was the work from the Puneet from our BK Modi Government Pharmacy College. It is contributed patent. So it was the study regarding the solid state physical properties improvement of cefuroxime exatil by particle engineering. Basically, this was the aim for the particular research study. Next, the method was selected in the study was the spray drying. Basically, the spray drying of suspension of excipients or spray drying of all excipients that is carried out, whatever desired carried, and we sprayed the dispersion from the upper side from the pump. You can see here that is a 5% slurry were used and 0.7 that is a nozzle diameter was 0.7 mm. Then we you can see in the diagram that our lab spray dryer, that is whatever at our college it is present. So basically that is normally we obtained a different preliminary batch results, and then another method also we adopted for the same to comparison, that crystallo co-agglomerates also prepared by the same drug and same co-processing for co-processing. So basically that in crystallo agglomeration we stirred that is by selected organic solvent insoluble drug we diluted with the excipients and the solvents, and then precipitation is carried out and agglomeration, whatever agglomerates we received, that is normally characterized for the different evaluations. So basically these were the material were used in the approach 1 and 2, that is normally spray drying and particularly agglomeration, crystallization. So DSC analysis was carried out for the particular basically our drug, selected drug. FTIR analysis were carried out. So this speaks that basically next sir, Chetan sir, next. These peaks were basically we obtained, which shows a particularly that resemble peaks were obtained, which it indicates that there is no chemical changes in the mixture and original drug. So basically this was carried out. Next sir. Next sir. Ketan sir. Ketan sir. Okay. So this is, you can see that DSC of the DSC thermogram of physical mixture of cefuroxime and different our excipients that were used in the particular study. There that peak shows that there is no changes in chemical structure of the drug with the particular drug that we selected all the polymers. Next sir. This is the method 1 for preliminary trials for T1 to T5. So there initially that different concentration of lactose, MCC, mannitol and chitosan was selected, and the parameter was set initially was 0.5 bar for atomization, and inlet temperature was 170 degrees Celsius that was initially kept, and by that initial trials we looked at different problems in the lactose. So we eliminated immediately lactose, and we used mannitol in all the different formulations, and later on that inlet temperatures and atomization pressure set and carried out the trial, which was promising for the future co-process excipients. Next batch. Next so T6 to T9 batch is also that is carried out with the drug as well as normally that is in case of the drug also that is taken. Next, sir, T10 to T13 batches were carried out for the drug as well as MCC, mannitol, with the setting the inlet air temperature 70 degree that was obtained was proper, and then that is carried out. Then the dried spray-dried product also characterized for the FTIR study, and that is normally found that there is no interaction between FTIR study in drug and as well as excipients. Then these are the pictograms of HPLC method developed for the spray dryer product. Analytical method is very much important to identify the co-process as well as the performance of the dosage form. Next, sir. So these are the design of experiments that is normally uh, uh, carried out. For that purpose, we selected a inlet air temperature as a independent variables and ratio of chitosan chlorhydrate and mannitol. The composition of chlorhydrate and mannitol was selected uh, and carry out the design. Next, so DO is applied according to that the particular combination. Next, sir. So here you can see the evaluation of design batches. So that design batches you can see that drug and different excipients that were used, and there this was evaluated for Cars index, angle of repose, Hosner's ratio, tensile strength, Kuno constants, and percentage friability and percentage yield. So all those were evaluated properly. So P9 batch was the promising batch for that particular purpose. Then Cars index, that is, you can see here that regression analysis model for the particular model. This is. Then you can see here that is normally that inlet temperatures and chitosan chlorhydrate and mannitol, that is how they affect particularly as the particularly ratio of chitosan and mannitol is normally as improved, that is the Cars index is lower, reduced as the Cars index is lower. So that you can see that that change according to that that we changed the formula and obtained a Cars index properly. Next, sir, this is the ANOVA table for the particular regression analysis. So next, sir. Ketan sir, next. Okay, so here you can see that basically that Hosner ratios analysis for the ANOVA table that found that is 37.34 F value calculated, and significant that is F tabulated received a 5.41. You can see. Next, sir. So here you can see this is a particular Hosner ratios comparison. Next, sir. Next, Ketan sir. So tensile strength, you can see that you can tensile strength also that is verified with the percentage of chitosan and chlorhydrate, and mannitol. Next, sir. So here you can see that Puno constant evaluated, the composition is evaluated for Puno's constant, and F calculated and F tabulated also, that is you can see here that is evaluated. Next, sir. So this is a Kuno constant that with respect to chitosan, chlorhydrate, and mannitol you can see. Next, sir. So basically that optimize co-process excipients according to the method one, that our goal where we achieved as a limit, as the limit is managing. So regarding inlet temperature and chitosan, chlorhydrate, mannitol, we kept in a range, fixed range, and we obtain a Cars index, we minimize Cars index and angle of repose, Hosner ratios, and then tensile strength maximize. So these are the results we obtain. Next, according to the method one, that all those things we desirability we achieved, and the area particularly we received for the checkpoint batch, and then it was carried out, and it was found that is good in the range. Next, sir. This is for the with drug. Earlier it was without drug. Now same thing that carried out with the ratio. And next, sir, the different run were carried out for batches, SDP 1 to SDP 9 batches, and we received a different percentage of ratios according to that. Next, sir. So here you can see that different batches evaluation with the drug, Cars index, angle of repose, Hosner ratio, tensile strength, Kuno constant, all those we received, whatever according to our desired property in the case of P9 batches. So this was promising for us because the yield, percentage yield was highest, as well as all the parameter were compromising. Next, sir. So here you can see the Cars index, basically relation between the Cars index, that ANOVA table for Cars index you can see. Next, sir, this is the chart that is a counter plot for Cars index. Next, sir. Slide number 50, sir. Slide number 50, 50. Okay. So that angle of repose also that is evaluated for the same composition, and that tab ANOVA that is table that have calculated values obtained. Next, sir. So that angle of repose that is we obtain, particularly in counter plot for angle of repose and against the chitosan, chlorhydrate, and mannitol composition. Then next, sir, Hosner ratio that is basically evaluated for particularly different ANOVA table you can see here. That next, sir, counter plot for the particular Hosner ratio that showing that. Next, this is the table for tensile strength that we received. In particular cases SD5, that is 4 batches. So that was the promising. Next, this is counter plot for tensile strength. Next, Kuno constant that is evaluated for the particular batch. That you next, this is counter plot for Kuno's constant. Next, so all the optimization carried out for the same as per the method one, that Cars index, angle of repose, and Hosner ratio, and Kuno constant minimize, and by tensile strength maximizing, that carried out optimization. Next, So here also that is a overlay plot suggests that checkpoint batches and that was carried out and that was obtained a desirable results. 60 number slide, sir. Next. So these are the comparison of optimized batch evaluation parameters for co-process that is prepared excipients and product, that is method 1 to both, that you can see that here table. Next, this is a radar plot that suggests car signals and angle of repose, particularly for drug, MCC, mannitol, chitosan chloride, right? Physical mixing, co-process excipients, spray dried products. So it suggests that some area that is a promising area from that particular region, that formulation is optimized. Next. So here some of the evaluation parameters is taken for the particular drug and carried out different evaluation: Washner ratio, hardness, total weight, friability, then hardness, percentage friability, disintegration time, then drug content. All those things are identified for the product. Next. Kawakita plot also carried out for the particularly this co-process excipients and their mixtures. Next, 64. So that Haeckel plot analysis carried out for the co-process prepared and that for py value that is obtained for co-process excipients and product is 2.123 on around 1.73 for product. Next. Dilution potential is very much important to identify for the co-process excipients. So that co-process excipients, that is a dilution potential carried out against tensile strength versus the percentage viability versus dilution potential. So it was a important criteria for evaluation. Next. Finally, the method 1 and method 2, according to that formula set uh, with the 250 mg that is cefuroxime tablets prepared and 125 mg cefuroxime tablet prepared, and for both that different formula that is decided with the co-process excipient, and according to method 2 that also same thing carried out, and this formula we again uh, carry out for the product. Next. Then this formula was finally the drug release check for the formula different. So that similarity index, dissimilarity index F1, F2 that is carried out, which was normally for under the range according to the requirement. Next. Short term stability studies were carried out for method 1 and 2, and these are the some of the parameters. There was no bias in the initial 0-day condition and the tested day condition. So it was considered that short-term study shows that it is a good in a stability. Next. Finally, that we can conclude that whatever co-process excipients we prepared, which was very good for dilution potential, Kawakita equation, Haeckel plot analysis, everything comparison, which was showing that the physical properties of the particular co-process excipient is more capable than the individual excipients. This is next. This is our college where this study were carried out and student filed that patent for the particular. So this is particularly very good vast area, because in the future there will be the demand for co-process excipients, because co-process excipients are reducing the many steps of the pharmaceutical manufacturing processes. So basically, this will be the company. Today, many companies are facing a lot of instrumentations, lot of processes do, compared to the direct compression. That example, weight granulation method is carried out. So there are processing steps are more in case of the direct processing. So there will be the ready mix type excipients. Just mix the drug with the particular excipients and directly compress will be the desirable in the future. So this area will be growing if anyone is planning for the business. So this is a very vast area for co-processing excipients. So this is all about today's presentation. If any query will be there from your side, you can write me in email. I will answer you. Yes, sir. Ketan, sir.
Sir, thank you very much for your very depth and detailed presentation about the co-processing. Sir, there is one small question that for which type of
Yes, sir.
pharmaceutical formulation, for which type of pharmaceutical formulation, this co-process excipients are more suitable?
Yes, sir.
Yeah, for which type of formulation this type of excipients are more suitable?
Yes, sir. Thank you very much. I would like to uh, say thank you very much for Principal Parul Institute of Pharmacy and Research, Dean of Pharmacy Dean of Parul University, and thank you very much for Parul University who give me the opportunity. First time I am delivering the speech outside the institute. So thank you very much for providing me opportunity for this such a wonderful presentation. Thank you.
So there is one small question.
Yes, sir.
Yeah. So for which type of dosage form this co-process excipients are more suitable? Is there any criteria for the selection of the excipients that in this particular dosage form it will be used?
You can use in any dosage form, and you can prepare a co-process excipients according to that for any dosage form. No, that is criteria, but you should have a desired target. What you want to achieve? That you wish to achieve a compressibility, you wish to achieve a flow property, you wish to achieve a disintegration. You are what is your target? So according to you can frame co-processing and you can prepare a co-process excipients. Sometimes multiple functions can be achieved by co-processing. Some excipients gives a compressibility, good compressibility, as well as good that is basically compressibility as well as good flow properties like that. So you can design in that way and you can prepare.
Okay. Thank you very much, sir. You have given a various example of the co-process excipients, advantages of the various excipients, along with the explanation of the case study for the research work that you have done for preparation of the co-process excipients. So on behalf of Faculty of Pharmacy, Parul University, I would like to thank you for your wonderful presentation. Thank you very much, sir.
Thank you. Thank you very much. Have a good day.
Thank you.
Is there anything, sir? Ketan, sir?
No, sir. No, sir. Thank you.
Okay. Thank you. Concluding the meeting. Thank you.